Hello, everyone. I'm glad to share our work in iMeta, and the title is Linking Soil Fungi to Bacterial Community Assembly in Arid Ecosystems. Soil microbes play important roles in a variety of ecological processes in terrestrial ecosystems. Revealing the fundamental mechanisms of biogeographic patterns is a key topic in community ecology. It is widely acknowledged that both deterministic and stochastic processes influence the biogeographic patterns of microbial communities. However, a problem occurred. What are the roles of biotic factors in driving community assembly? Accordingly, we scientifically hypothesize that 1. Biotic interactions between microbial taxa contribute to substantial variations in community beta diversity across different habitats at large scales. 2. Soil fungi mediate the balance of assembly processes of soil bacterial communities. Based on the assumption, we collected 251 soil samples from different ecosystems along the Heshi corridor in the northwest of China to estimate diversities of bacteria and fungi. Our study focused on the biogeographic distribution and community assembly of soil bacteria. As a result, the bacterial alpha diversity showed a greater value in low-latitude and high-longitude fields. Then, we explored the species colored the higher abundance of the soil microbiota via establishing cross-kingdom co-occurrence networks including soil bacterial and fungal taxa. We found that the degrees were significantly decreased with the increasing of aridity index, AI. Subsequently, across all abiotic and biotic factors, fungal richness, average degree, AD, and AI contributed the most toward explaining variation in bacterial alpha diversity of different habitats. This result was confirmed by significant and negative simple linear regressions found between AI and bacterial alpha diversity. The strongest negative effect of AI on bacterial alpha diversity was observed in desert soil, and bacterial alpha diversity were more affected by AI at the subsurface layer. The major abiotic and biotic factors to the bacterial beta diversity were further identified using the CAP. AD and AI were most important predictor for the soil bacterial beta diversity. The slope of distance decay relationship, DDR, in the agricultural soils was significantly steeper than the other habitats, and the slope in forest soils was the flattest. We combined multiple regression modeling and variance decomposition analysis to quantify the contributions of the factors to each dominant taxon. The results showed that AD was the most frequent of the best predictors of their abundance, followed by AI. Significantly more explained variations were observed when considering the biotic factors rather than only considering the abiotic factors, indicating the major roles of biotic factors in governing the bacterial assembly. Null model analyses revealed that the deterministic assembly was dominant in bacterial communities in agricultural, forest, and grass soils. The relationships between T and major factors were used to infer changes in the relative influences of deterministic and stochastic assembly processes. Fungal richness was the best predictor of T. With increasing in fungal richness, the relative influence of stochastic assembly processes of bacterial communities decreased. To provide a further test of the importance of fungal richness for bacterial community assembly, we conducted a manipulative microcosm experiment with independent soil samples. We experimentally create a gradient of soil fungal richness by using fungicide in independent soil collected from grassland. We found fungal richness significantly decreased with the increase of fungicide concentrations, and stochastic assembly processes of bacterial community increased along with the decreasing in fungal richness. Besides, bacterial beta diversity significantly differed among the fungal richness gradients. In conclusion, through field sample collection and laboratory experiments, we revealed the key role of biological factors in driving the formation and construction of bacterial communities. Soil fungal richness mediates the balance of assembly processes of soil bacterial communities, with stochastic assembly processes decreasing along with the increasing in fungal richness. Increased aridity conditions due to climate change will reduce the bacterial alpha diversity, with more negative effects in subsurface layers, and induce more antagonistic or competitive biological interactions. Thank you for your attention. iMeta, a journal to change the world.